Oh, sorry, Chris, I had on mute, but we're now starting class, so you should be able to hear me now.
Okay, I can hear you. Thank you.
Okay, 8.2 today. So we talked about last week about the experiments that look to see what is being transferred from parents to offspring. Is it protein? Is it DNA? And the experiments confirmed that it is DNA that's being passed on. So after they knew that, they did more experiments later to understand what chemicals make up DNA, how is it structured, how do those chemicals affect what's being passed on, how do those make genes. What they discovered, and they were really amazed by this, is that DNA is the same structure in all organisms, whether you're talking about bacteria, plants, animals, humans. It's the same exact chemicals and the same exact structure. No matter what living animal or human or plant you're looking at, it's all the same structure. Now it's arranged in different sequences, and that sequence gives you what a human versus an animal versus a plant cell, but it's still the same chemicals in each. Does everybody have this slide down? Okay, so let's go through the parts that make up DNA. Scientists discovered that there are 4 nucleotides that make up DNA. And DNA is just a long chain of nucleotides. For a human, it's 3 billion, billion with a B, nucleotides long. So scientists knew this for a while. They knew that there are 4 nucleotides that make up DNA, and they assumed it was just too simple to be genetic material. How can you have such a wide variety of traits in humans, animals, and plants if all you're dealing with is just 4 chemicals, and just the variations of chemicals in their sequence gives you the different traits? But they learned that it is enough to make all the different things we see in humans versus animals versus plants. There are 3 main parts to the nucleotides, which you can see the diagram down there. So you should draw this diagram too and understand these parts. I'll go through each of them. So the first part is a phosphate group. That's what you see as a circle right here. And then attached to it, the pentagon right here, that's a sugar. It's called deoxyribose. Yes.
Is that why it's called deoxyribonucleic acid?
Exactly right. Yes. Yeah, because and then RNA is called ribonucleic acid because it has a ribose sugar instead of deoxyribose. Yes, the name tells you what's in it. And then you have a nitrogen-containing base, which you might see as a pentagon and a hexagon, as you see right here, or 2 hexagons. So those are the basic parts of DNA. And that just makes up 1 nucleotide. And you have about 3 billion of them in each cell in your body. And the sequence of those gives you your traits that you have. Does everybody have these down? So whenever you see it in chemistry or in biology, if it ends in OSE, it's a sugar. So certain sugars that you see, lactose is sugar you find in milk. Fructose, That's sugar you find in fruit. And so that's a Latin word, fructose comes from a Latin word for fruit. And so that's fruit sugar. Sucrose is table sugar. Glucose, blood sugar. So if it ends in OSE, it's a sugar. So let's go through the 4 bases. So the nitrogen-containing bases are the only difference in the 4 nucleotides. And so you can see, going back here for 1 second, what's in green right here is what changes. And so that would be the part that's variable. So some of them you can see it's just one hexagon, like thymine is just this hexagon right here. And so that would go in place of the green pentagon hexagon you see over here on the other slide. Cytosine, also hexagon. So these are single rings. And the double rings would have a pentagon and a hexagon together. Yes.
We need to draw those or just-
You don't need to, draw all of this right here, but you should draw in the hexagon or the hexagon and pentagon. So you can ignore the structural formulas, but you should draw in the model right here you see and the name of the bases. So there's 4 main bases. You have thymine, which is a hexagon, cytosine, which is a hexagon, adenine, hexagon and pentagon, guanine, hexagon and pentagon.
What was the picture before? I mean,
The picture before,
it
it could be either. I mean, green, it doesn't really mean that it's actually green. And so it could be either of these, guanine or adenine.
But what I mean is like it won't always look like that. It might only have one thing.
Wait, sorry, say again?
Those 2 won't always look like that. They might have one thing.
Well, so it's either adenine or guanine. Because this right here has a pentagon and hexagon, right?
yeah, I'm saying is sometimes it won't always have those 2. It won't always look like that. And that's just one.
Oh, well, yeah, because the one that's just one, that's thymine or cytosine, right? So that would be either. But between the 2, we don't know if it's adenine or guanine, but it'd be one of those 2. So this is basically the code of DNA. Whether it's a T, A, C, or G, that's the language that it has. And that gives you the different traits you see. If you could write all the information you have in your DNA in just one single cell, you'd have 1,000 books, each 1,000 pages long of information. That's how much information DNA holds in each cell in your body. Does everybody have this one down? Okay. So our alphabet is 26 letters long. DNA's alphabet's only 4 letters long, but even still, it's still able to have a lot of information that it can hold within those 4 letters. They can do that because it's so long. I mean, it's billions of letters long, and so it can have a wide amount of possibilities because of how long it is. So Watson and Crick in the 1950s determined that DNA is double helix. So they looked at the 3-dimensional model of DNA and they determined what DNA would look like. And they call it the double helix. You've all seen that before. You have the 2 strands intertwined. So it's made of, basically, we looked at the 3 parts of DNA. We can actually simplify it into 2 parts. The sugar phosphate backbone, which would be the deoxyribose, along with the phosphate group, makes the blue ribbon you see along here. And so the blue ribbon contains the phosphate group and the deoxyribose sugar. Then the other part to it, looks like the ladder rungs right here, these rods that come through it. Those are the bases, adenine, guanine, and cytosine, and make up these colored bases right here. And that's the part that varies from person to person, from species to species, would be the kinds of bases you see along here in their pattern, whether it's adenine, guanine, cytosine, or thymine, that's what changes. And that makes you and different from other people. So that's Watson and Crick, and that's their model of DNA. They use wood and metal to create this model. So you might wonder, how did they see this? Because you can't actually see a single strand of DNA. We saw DNA in the laboratory last year, but it wasn't just one single strand. It was many strands put together, clumped together. So to see one strand is impossible. We can't do that. We don't have any microscope that can look at a single strand of DNA. So they actually had to use X-rays to see what it would look like. And they used X-rays of somebody else. They used Rosalind Franklin's X-rays of DNA and Erwin Chargaff. They worked together on this. And what they did is, by shining an X-ray on DNA, they got this right here, this X shape. And that X shape is what told them that DNA is in a double helix. So they had to extrapolate based on the X-ray images, and those X-rays told them that DNA had to be a double helix shape. But that's what they had to deal with. Yes.
So what are they looking, what are they looking at in that picture?
It's a top-down view of DNA. So this is an X-ray image, and the DNA scattered the X-rays, and it scattered them in such a way that it shows this double helix formation. So think of it as like a single slice. Like if you were to slice it top down, that's what you would see. So Franklin's X-rays images are what they used to show that DNA was double helix. They stole them from her. She didn't give them permission to take them, but one of her colleagues gave it to Watson and Crick, and that was what they needed to know to make their model. And so she already knew about DNA being double helix. She hadn't actually written a paper about it yet, but she knew that that was what it was showing her, but they took credit for it. Watson and Crick took her data, stole it, and they won the Nobel Prize because of it. And she didn't, because they didn't even give her credit for her discovery. Of course, now we know that she was the one who discovered it, and so now we do credit her for that. But at the time, the Nobel Committee didn't know that. They don't give Nobel Prizes to people who are already dead, and they only realized this after she had died. And so they didn't award her the Nobel Prize because during her life, they didn't know that she did this. Aaron Chargaff was working with Rosalind Franklin, and he realized that adenine pairs with thymine and cytosine pairs with guanine. That in every sample of DNA, the amount of adenine equals the amount of thymine, and cytosine equals guanine in amounts. And because those are the same exact amounts, what he said is that adenine must pair with thymine in the double helix, and cytosine with guanine. So that's called Chargaff's rule, which says that adenine equals thymine in number, and so they must pair together. Does that make sense? And so what he said, like in an individual's DNA strand, he might count 40 adenine. He also would have counted 40 thymine. There might be 20 cytosine, but there also be 20 guanine. So the amount of cytosine number always equals the guanine number. Adenine always equals thymine in number. And based on that, he said, well, adenine must pair with thymine. So Chargaff said that nucleotides must always pair the same way, because his data analysis of DNA always showed this to be true, that adenine always equals thymine in number. And so adenine must pair with thymine like this. You know, where you see it on the ladder rungs on the DNA, A goes with T and C goes with G. So A and T always pair together. C and G always pair together. And this makes sense when you think about the structure of cytosine, adenine, thymine, and guanine. Because remember what we talked about before, thymine has a single hexagon, adenine has a hexagon and a pentagon. So adenine is bigger than thymine. Guanine has a pentagon and a hexagon. Cytosine just has a hexagon. So like puzzle pieces locked together, you know, you see that these each together make up the same exact amount of length. But if you had guanine pair with adenine, it would be too big in some places and bulge out. Other places would be too small. But pairing up this way, it always ensures the same exact amount of spacing between the two nucleotides. So single rings pair double rings, and that makes a uniform helix shape. Now, hydrogen bonding is a bond that's pretty weak. Covalent bonding is pretty strong. So it's kind of like, you know, like a Velcro, where you need to have it stay together when you want it to, but pull apart when you want it to as well. DNA is like that as well, or like a zipper. You know, you need to be able to pull it apart when you need to, but also keep it together when you want it to. So you have to pull apart DNA when DNA is duplicating to make more cells. Otherwise, you want to stay together, because if it stays apart, it's more prone to mutations and damage. So you want DNA to be able to stay together when you want it to, but to pull apart very easily when you want to duplicate it. So hydrogen bonding accomplishes that. It's a Velcro-like bonding that is easy to pull apart when you need to, but sturdy when you want it to stay together. Covalent bonding is a very strong bond, and the backbone always stays intact. So the backbone is connected by covalent bonds, which you should write down is a very strong bond, which is very hard to break. And so the backbone is not easily broken, which is important because you would have mutations at that point. If you broke the backbone, you'd have the bases out of order, which would cause mutations to happen.
So is that possible?
Oh, it definitely happens. UV radiation can do that just from the sunlight. Chemicals can do that. Physical damage, you know, if you cut your skin, you know, you smash cells, those are all things that can damage the backbone. And so that can and does happen. And the cells can die from that, or they can just be mutated, and that can cause things like cancer to form as well, too. And hydrogen bonding in the middle, like we talked about, also, I would write down that that's a weak bond. It's a bond that, just like Velcro, is weak and where it needs to be weak, and strong where you need to be strong. And so it accomplishes both strength by keeping them together, but also flexibility, and you can pull it apart when you need to copy the DNA. And so this pattern of nucleotides is what makes you you. It's what gives you your genes. And the only person who has the same genes as you would be if you have a twin who's identical to you. Besides having identical twins, these are all going to be different, not all, but mostly different from person to person.
So where would the genes be in all this?
The genes would just be a segment of this. So it'd be about 1,000 or so, maybe hundreds to 1,000 or so of these nucleotides. It makes up a gene.
So, like, those are part of.
That's right. Mhm. So the unique formation of these, the unique arrangement of them is what gives you a certain gene. And so there's a certain code, like TACCCAG, and so on. That whole code would tell you a certain gene is present. And so when you do genetic testing, like if you 23andMe, it's reading these letters right here. And based on those letters it's reading, it tells you what genes you have. Okay, so that's 8.1. Today I want you to work on two things. Sorry, that's 8.2, not 8.1. 8.2. So today there's two things I want you to do. There's homework questions on page 219, 1 through 6. And then go on to the next page after you've done that, on page 220, and do the data analysis lab, questions 1 and 2 on page 220 after you do 8.2. Yes,
You put it all together on the paper.
that's fine.
Miss Holly,
Yes,
could you send me the questions for those?
yes, I can do that.
Thank you.
You're welcome.
Would you also send me the questions?
Yes, I can do that.
Okay, thank you.
All right, have a good day, Kristen and Jordan.
Thank you.
Thank you.